Okay, hello everybody. I'm going to go over the software that we'll be using in the course for analyzing gene sequences. First, I'll show you where the website is to sort of access and discover DNA or gene sequences, and then we're going to look at a software program called Mega, in which we'll use to analyze those sequences. So this is intended to be a very brief introduction to kind of just get you started. We're going to go into a lot more detail about what the software is actually doing and the proper way to use it, but I just want to kind of get everybody started off on the same page. So first you're going to go to a website called Ncbi. This is the database for the National Center for Biotechnology Information. It is also houses a database known as GenBank. GenBank submissions are done whenever DNA sequences are published, and these are made public by the U.S. government and are available to everyone. Now, the Ncbi website is very complex. There's actually a lot of databases here. On this drop-down menu, you can see a list of them. The only ones we're going to be very concerned with are homologene, which are homologous genes. I'll explain what homology is in a minute. And then these are all sort of centered around gene pages. So if, for instance, if I went to the gene database and I wanted to look for alcohol dehydrogenase number 4, and I do a search for ADH4, I can find alcohol dehydrogenase gene in humans, and then I can also go to this page and see that gene sequence, for instance, here. So at the heart of the Ncbi database is essentially pages like this for every gene, over 20,000 genes in the human genome. More and more these are being discovered by automatic gene finding algorithms. So in the case of those genes, they're often called predicted. If you see the word predicted in the header, that means that it's probably not been curated. It's just been sort of predicted by an algorithm, and it's not being curated by people. In the early days, a lot of the genes were curated. Now, if I go back, there was also a link to orthologs. So these are also sort of a form of homology or a homolog sequence, and I can find in this this is a relatively new thing they've added to Ncbi, this Ncbi orthologs page. But you can see here that I can actually go here and I can click and sort of collect the human sequence, the mouse sequence, the sequence for cattle and rhesus monkeys, etc. And then I could actually download those into a FASTA file. Now this is something that you can use in Mega software if you were to download these. But I'm going to, I find this ortholog finder can be a little bit overwhelming. It's a little bit quicker if you wanted to grab like hundreds of sequences. But if you're only going for 7 or 8, I sort of like the homologene database, which is a little bit older. So either way you do it, I'm going to go back and sort of show you that database right now. So let's go back to the front of Ncbi. Now, before we go forward with this, I want to tell you that when you're in Mega, you're actually able to open up Ncbi and download and bring your sequences into the Mega software platform. So it's actually better to do all of this within Mega. I haven't even shown you Mega yet and haven't opened it yet, but I'm just sort of showing you about how you would sort of navigate Ncbi once you do that. So again, if you go to the databases and you go down to homologene, I can click on that, and I can also search ADH alcohol dehydrogenase number 4. And now I have 3 search hits here and it sort of tells you, that these are conserved. So the first one is you're going to have to learn a little bit of Latin. Unfortunately, biology uses a lot of Latin terminology. So Ascomycota, that if you're really aware of your taxonomy, you would know that that's a cup fungus. That's a type of fungus. So this is ADH4 that's only in fungus, and if I were to open that, I've only got like 3 genes here from a few different kinds of fungus there. So I've got a yeast gene, some other, and a couple other yeasts. So this isn't that interesting. If I go down to this one here, it says conserved in Eukaryota. Now this eukaryotes are all essentially multicellular organisms, including yeast. So if I follow this one, I can find that now I have a whole set of sequences here that I can go after. And in this case, I have a human sequence. Pan-troglodytes is chimpanzees, macaques, the wolf or dog, cattle, mouse, rat, and X tropicalis is xenopus. This is actually the African clawed frog that you buy in the pet store, but it's also a model organism for genetics for amphibians. Arabidopsis thaliana, that's the model organism for plant genomes usually. And then there's also Orris sativa, that's actually rice. So you might find crops and things like that. You can see that this protein is relatively the same. It's one large domain, and so therefore there's this colored box. If there were more domains, it would have breaks and different colors here. If I follow these sequences, this would take me to the human protein. NP and XP usually denotes a protein sequence, and a protein sequence might be something that we would want to download, and you can see here that at the very bottom of this I have again the 20 letters that indicate the different amino acids in this protein. So this is the protein sequence. Remember, they almost always, all proteins start with methionine, so they start with M, and that's the protein sequence. Now if I go back over here I can go to the gene page, and here I can get either the DNA or the RNA sequence. In this course, I'm going to encourage you to mostly work with messenger RNA sequences, and this is because of the simple fact that in eukaryotes, the mRNA is essentially very close to the coding sequence of the gene usually. It's because all of the intron sequences have been spliced out of the messenger RNA. So, so where would you find that? If you go down the page here, if you can find a coding sequence, or a CDS as it's termed, that's usually the best thing to get if you can. That's the DNA coding sequence. But in lieu of that, sometimes this NM here, which you'll see an arrow and then a protein. So this would be the protein sequence, and this is the messenger RNA sequence. If you get other DNA sequences in this Ncbi reference sequence or ref sequence download, some of these can be quite large. Like if you just pulled down, a lot of times sequencing projects have very large contiguous sequences or contigs that are assembled, and these have many, many genes on them. If you pull them down in Omega and try to analyze them, at the gene level, you're going to have very huge sequences that take very long periods of time to align. If your alignments are taking really long time, it's a sign that you've pulled down sequences that are way too large. And what you really want are probably these mRNA sequences. So mRNA, this is the messenger RNA for the alcohol dehydrogenase gene. And within that, it will have a DNA sequence here at the bottom. Within this sequence will be the coding sequence, the exon sequence. There's also a 5', what we call a 5' UTR, or untranslated region, and a 3' untranslated region. So there's usually some trimming that needs to be done, and I'll show you how to do that in Mega in a few minutes. So let's talk about Mega. I've shown you the sequence where you can find the website where you can find sequences, but you're not generally going to work from here. You're going to start with a program known as Mega that's installed on your computer. You can use Mega X, or you can use version 10 for Windows. or some of these other versions. Mac versions have not always been very reliable, but the Mega X is cross-platform, so it should be it should work pretty well. Choose the graphical interface version. You can also do a command line version. This is useful if you're already a programmer in the course. You're coming over from computer science, you know how to program. The command line's nice if, say, you wanted to do analysis on not just one or two genes, but hundreds or thousands of genes, then of course you can do this. You can actually call Mega and use it in loop structures and things like that. And that's beyond the level of this course. We're going to be working only with the graphical user interface version. So when you've installed Mega, it'll give you an icon like we have down here. And this is what Mega looks like when we open it. So, so let's talk about the steps to analyzing gene sequences in Mega. The first thing that you're going to need to do is it's always a two-step process. The first step is to create an alignment of sequences. We'll talk more about this in the class, but basically an alignment is a procedure, an algorithm that we use to enforce homology at every site. So that means that if I'm looking vertically up and down my sequences in my alignment, I know that that vertical column is the same site on each gene, and therefore I could compare the states of A's, T's, C's, and G's at those sites. I can't do that if I don't have homology, and I can't do any analysis of mutations between species or between different genes if I don't have that homology. So alignment's very important. That's the first thing we do in Mega. The second thing we do in Mega is analysis, and that's what all these other buttons are for. So, and generally when you work with Mega, it's always best to create an alignment and export an alignment file, close Mega, and then reopen it as your data file. And the reason we want to do this is there's no point in doing all of these analysis here unless you have an alignment that you're confident with. So Mega sort of makes this as a separate two-step process. The first one is obtaining your alignment, and the second one is all the subsequent analysis you might do after the fact. So when you go down to Align and you go to Query Databanks, you're going to see that Ncbi opens right up. But now you're inside Mega. You're in the Mega web browser, and you have this little button here called Add to Alignment. So for instance, if I were to go back to my my homology search, and I search for ADH4, then I can go back to this page, and now I can go to that mRNA sequence. So I go down to where it says refseqs, and I look for that link here, NM or XM, and that takes me to the messenger RNA page. Now, if you have, this is essentially a web scraper tool, so if you have sequence on this page and it's recognizable, I can click Add to Alignment, and it will pull that out automatically, and it should put it right into my alignment in Mega. So here I have my sequences. You can see them here. So I've pulled down the first sequence, and now I can go back and I can so I can walk backwards, and I can go for the chimpanzee sequence here. And I can do the same thing. I go down the gene page. This is all the gene annotations. I look for the mRNA and protein. I find my mRNA here. It's labeled XM, and I pull that one. And then, if you have sequence on the page and you press Add to Alignment, it will bring the next sequence in. So there we have the next sequence. And I'll go back and I'll grab one more, And then I'm going to talk about alignment. Now, typically you would grab like 8 or 9 or 10 or 12 sequences for a multiple sequence alignment. And I'll open that up and sort of show you how to analyze that. But just for the sake of actually running the alignment function, I'll go ahead and pull one more sequence in. And there it is. So you can see that there are some sites where there are differences and some sites where there are not. You can see the human and the chimp sequence are nearly identical at the level of DNA, but this sequence I have from cattle or Bos taurus is quite different in different places. Now I can go back with these sequences. I can actually translate them to protein if I wanted to. And this is the protein sequence. I can go back to my DNA sequence here. And I can also create an alignment now. These sequences, you'll notice, may not be exactly the same length. In this case, they are. But in many cases, the gene could be shorter in one species than another. And when I look vertically here, these sequences are not necessarily homologous at these sites until I run the alignment algorithm. Now there's two alignment algorithms in Mega. There's one called muscle and another one called clustal. We'll talk later about sort of the difference between these. We prefer clustal generally, aligned DNA. And then we would click this and that will run very quickly and sort of impose an alignment in my sequence here. Just for the sake of maybe demonstrating this alignment concept a little more carefully, I'm going to go back and get a much more distant sequence from here. Why don't we go ahead and get the Arabidopsis sequence. So this has evolved to be very different over time. So now you can see that sequence is really quite different, and this is where an alignment would be very useful. You can see there's a lot of mismatch here with this other sequence, and so an alignment here would be really critical. So now I've run an alignment, and you can see that it actually puts some gaps in. So it's actually gone in and tried to use gaps and other things to try to align these two, this third sequence with the other three. You can see there are some places where it is aligned fairly well, and there's other places where it's aligned not so well. So it's always very important to assess your alignments visually. In this case, there's the level of mismatch is still relatively high, so I would sort of question whether I would want to keep this in my analysis or not. It would really depend on the type of analysis I'm doing. But anyway, that's the sort of way that you generate an alignment. Once you've created an aligned sequence in this Alignment Explorer page, you're going to want to go down and then click on Data, Export Alignment in Mega format, and then save that to your desktop. And once you've done that, then you can go ahead and close Mega down. And to sort of show you now what you can do with sequences. Here I've created a much more, larger alignment, and we'll go ahead and maybe do a few analyses on that. So here I've actually created an alignment. I saved it as an alignment file, and now I've just double-clicked on it, and it opens up in Mega. I can see my data in this alignment. Now I have 10 different species aligned. You can go in and You can color your cells to sort of look at that alignment here, but you can't go back and realign things at this point. And the reason why Mega doesn't let you do that is it doesn't want you doing a bunch of analyses and then potentially alter your alignment and not realize it. And then you have sort of the garbage in, garbage out problem. So once you have an alignment file that you are confident you want to do your analysis on, you open that up, and it will always sort of be sitting here behind this tab. But we can do a lot of different kinds of analysis. Now let's talk about these. One of the first things you might want to do is a phylogeny. We might want to generate a tree. And there's many different methods here. We're going to talk about them in the course of this, in the course of the class. But I'm going to show you just how quickly and easy it is to do. Let's do a neighbor joining tree using the active data set that I have open. There's a lot of settings for these different things. I can go in here and I can ask for, say, a test of phylogeny by doing bootstrapping. So I might do 500 different phylogenies and I ask how similar are they when I run this algorithm. I'm not going to do that right now. And then there's lots of different sorts of models and methods we can choose from. But we're doing a neighbor joining tree. Neighbor joining tends to run very quickly. It's a more primitive algorithm, but it's one that we often use for really large data sets even now. So here you can see that my plant sequences are much further away than my, you can see that my human and chimp sequences are almost identical, so the branches are almost non-existent here. Very close to monkeys as well. The other mammals are all very close. Notice that the mouse and the rat are actually quite distant compared to other mammals. In fact, there's a bigger difference. Rats are not just large mice. They're actually quite a bit, what we say is diverged evolutionarily, much more than primates are. And you can see that here. We have an outgroup with our plants and then again with our amphibians. So our tree makes sense, even with this relatively simple algorithm called neighbor joining. There are other types of analyses we can do. The gold standard for phylogenetic inference is maximum likelihood, and there's a lot of different sorts of models here that I can choose from. For my substitution model, I can choose, I can do my analysis on nucleotides, and then I can choose a whole different host of types of models here. We're going to talk more about what these different models are. The natural question is, which one should I use? Which one is best? Jukes-Cantor is a very simple model. The GTR is a very complicated model in terms of the number of parameters being fit to your data. If we jump, I'll go ahead and choose the simplest one. But that question of which model to choose is important. All of these DNA substitution models are based upon something that looks like this. If we look at a Markov, they're actually called linked probabilities or Markov models. And you can sort of analyze DNA sequence by saying, what's the probability in my data of anywhere a T changing to a C? And that would be defined by this parameter, the rate of T changing to C. I could assume that all of these rates of change are equal to each other, and that is in fact the Jukes-Cantor model. It assumes one rate of mutation across the whole thing. But I can also say that in my data it might be much more common for mutations to occur in one direction than another. And so that's what all of the differences of those various models sort of indicate, or the way that these are set up. There's always a balance between Occam's razor having a simple model versus a more complicated model that might overfit your data. Overfitting is when it explains your data set really well, but the minute you give it other data, it doesn't perform well because it's already overfit or overtrained on your particular data. So often simpler models are much more explanatory. There's always a balance between simple explanation, Occam's razor, versus a more complicated model that might be fitting too much noise. in your data. We'll talk a lot more about this in the course of the class. But if you look up Markov models of DNA substitution, you can kind of see these, and we'll talk at much more length about these in the class. But that's a model that's actually being fit to our data in Mega. So when we choose the Jukes-Cantor model and run it, and there's other types of parameters we'll talk about as well. In this case, I'll go ahead and run a bootstrapping method, and I'll maybe do bootstrapping about, maybe we'll only do it about 50 times just to, in the interest of speed. So you can see that this bootstrapping of the tree is much slower. Maximum likelihood algorithms are generally slow. On a fast piece of hardware, though, it used to be that 20 years ago people would wait overnight for this thing to run. But now you can see I've already run it up to 15, 16, 17 bootstraps already. With what I'm trying to do here is I'm trying to infer, based on rates of evolution happening in my sequences, in my alignment, what that phylogeny might be, what the best fitting model is, given, or what the best tree topology is given the model that I've chosen. I'm going to show you something in a minute in Mega about how to actually choose the best model. But we'll go ahead and let this finish running here. It's almost done. And this will run a phylogenetic tree. And there it is. And we actually have numbers now that tell us the confidence. So there's not a lot of confidence, this 44, in placing this particular branch of the tree. But we have a lot of confidence everywhere else, 90, 100% in other cases. And you can see that this tree looks fairly similar to the neighbor joining tree. So we didn't really learn a whole lot more with these 12 sequences by doing this much more sophisticated method using maximum likelihood statistics than we would have just doing simple heuristic neighbor joining. But you can see it's very easy to just run Mega and get results and do them. But you really got to be careful about the models you choose and why you choose them. There's a section here before phylogeny. There's this button called models. And when it says here that find best DNA and protein model, this is very important. This is actually using, we'll talk about this more in class, using a method, another maximum likelihood method called multi-model inference, or Akaike information, or Bayesian information criterion is what it's sometimes referred to. But if I actually just run this, it will actually run through all of those models. And so that JC plus gamma, that's Jukes-Cantor. And it will give me a table that will actually tell me which ones are the best models to fit. So the models towards the bottom of the table don't fit as well as the models at the top. So in this case, it says, this is a K2, which stands for Chimera 2-parameter model plus a gamma correction, is the best model for my alcohol dehydrogenase gene. So that's the model I should select when I go back into phylogeny here, and I run a maximum likelihood tree. I should actually look for a Chimera 2-parameter model, and then I should use the gamma distributed parameter for the rates among sites. And that right there would actually give me potentially better phylogeny than other types of models. So, so it's important to run the model selection here before you run the phylogenetics. Some other things you can do, we'll talk a lot more about some of the other things here. I can actually do a distance matrix. This would actually calculate all of the pairwise distances between my sequences, again based on these rates of substitution, and these are there, you can see them now. These distances here are numbers that sort of reflect the distances between my sequences. These distances are actually what are used in in the neighbor joining heuristic algorithm. I can recreate ancestral sequences. I can infer ancestral sequence states here as well. This is pretty cool because it's a way of actually generating. Once I've generated my tree, I can actually go back and I can generate what the ancestors on that tree looked like. So I could go back with enough sequences, I could go back and recreate a Tyrannosaurus rex sequence. I could take that in the lab and make the protein, and people actually have done that. You can study ancient proteins by using these types of methods. Selection. This is for tests of selection. Again, there's a lot of sophistication here. The simplest one, the codon-based z-test of selection. We'll talk about this one in class, but this will actually tell us if there's significant levels of selection occurring on my gene or not. You really have to make sure you have a correct alignment here. These negative values of the z-test are actually, what these are actually telling me is that there's strong levels of purifying selection on this gene. So what that means is that, and these are the p-values or the probability values associated with that down here in yellow. The yellow indicates that it's significant. So what this says is when I compare sequence 8 to sequence 2, or if I compare the chimp sequence to the frog sequence, I get a -15. That means that the rate of substitution on the silent sites in the genome, the ones that don't affect the protein level, are 15 times faster than they are in the protein affecting genes. So what that means is the protein's very important and it's not being allowed to change due to random chance. If you had a broken gene, something that we call a pseudogene or a gene that is non-functional anymore, these rates become very equal and these numbers are no longer negative. So this is a way of actually testing the importance of that gene. It tells you it's a very important gene. It's very critical to our life, and evolution has not allowed it to change very much. The last thing I'll show you today is the diagnosis gene. This is pretty cool. This is a machine learning. Algorithm. It actually connects to a database that's stored at Temple University, where the makers of Mega are. I used to work actually for my center director at ASU is the creator of Mega, and he was working on this when I was at ASU. This is pretty cool. It's this is a method of essentially has an alignment. So if I go back to my ADH4, I'm actually calling the ADH4 protein for the human genome. I'm actually going to call up a multiple alignment for that. So here's the gene number, and if I actually say now I want to diagnose variants of that gene, protein variants. Now I've pulled up in my sequence explorer here. I now have the human sequence on the top, and then I have all of the other alignments. So this is a very large alignment that's been created at the protein level and stored in Temple. And one of the things it does is I can go back and I can make predictions about if I were to alter a particular protein in a particular place here, I can actually probe that. So So in this In this alignment, I can look for conserved sites. I can look for I can look for variable sites, and I can go back and I can analyze using machine learning algorithms. I can find out if certain particular changes to that amino acid sequence are detrimental or neutral or not to the functioning of the protein. And that's predicted by using a very large database of essentially a database of mutations that are known to have sort of medical conditions or medical information related to them. So it's a tool we'll talk about more later in the course. So that, in a nutshell, is MEGA, and it's the primary software we'll be using to analyze genes at the sequence level, the level of DNA sequence. We're going to be using some other software for analyzing protein structures, and I'll make a separate video to describe that. Okay, I'll see you later.